Today we will explore the anatomy and physiology of the skin through the lens of a hypothetical melanoma case study. We will begin by reviewing the normal structure and function of the skin, including its three primary layers, the epidermis, dermis, and hypodermis, along with its key accessory structures. We will then examine how melanoma originates in melanocytes within the stratum basale, its pattern of progression through the skin layers, and the resulting impact on critical physiological processes. This case underscores the clinical importance of understanding normal skin histology and highlights why each detection remains essential for optimal patient outcome. We'll begin with a quick overview of healthy skin. The skin is made up of three major layers, the epidermis, the dermis, and the hypodermis. Together, these layers protect the body, help us sense our environment, regulate temperature, produce vitamin D, and maintain fluid balance. The skin also contains important accessory structures like hair follicles, sebaceous glands, sweat glands, and nails. All of these components work together to keep the body functioning and protected. Now we'll look more closely at the structure of each layer. The epidermis has 5 layers, arranged from superficial to deep. The stratum corneum is the tough protective outer layer. The stratum lucidum is found only in thick skin, like the palms and soles. The stratum granulosum helps form keratin and waterproofing. The stratum spinosum provides strength and contains immune cells. And the deepest layer, the stratum basale, contains melanocytes and is the site of continuous cell division. Below the epidermis is the dermis, which has 2 layers. The papillary layer, containing capillaries and sensory receptors, and the reticular layer, which provides strength through dense connective tissue. This is also where we find hair follicles, sweat glands, sebaceous glands, nerves, and blood vessels. Finally, the hypodermis is made of adipose and areolar tissue. It insulates the body, cushions deeper structures, stores energy, and anchors the skin. We'll begin with our hypothetical clinical scenario. In this case study, we're examining a 46-year-old patient who presents with a rapidly enlarging, irregularly pigmented lesion on the upper back. The patient has a history of frequent sunburns and tanning bed use, which increases the risk of melanoma. Dermoscopy shows asymmetry, irregular borders, color variation, and a diameter greater than 8 millimeters. A biopsy confirms superficial spreading melanoma that has begun invading the upper dermis. The underlying cause is UV-induced DNA damage to melanocytes. Melanoma begins when UV radiation causes DNA mutations in melanocytes located in the stratum basale. These damaged cells start to proliferate abnormally. The tumor grows both radially across the epidermis and vertically downward toward the dermis. As this happens, the immune system responds, and cells like Langerhans cells and macrophages become activated. Once the melanoma reaches the dermal blood vessels, the risk of metastases increases significantly. In the epidermis, the earliest changes occur in the stratum basale, where melanocytes become dysplastic and begin spreading upward and outward. As the melanoma progresses, it invades the papillary and reticular layers of the dermis. This is especially concerning because the dermis contains blood vessels and lymphatic channels that can transport cancer cells to other parts of the body. Accessory structures like hair follicles, sebaceous glands, and sweat glands may be displaced or destroyed as the tumor grows. In more advanced cases, melanoma can extend into the hypodermis, affecting subcutaneous fat and deeper tissues. Under normal conditions, the skin regulates body temperature through vasodilation, vasoconstriction, and sweating. When the body needs to cool down, blood vessels in the dermis dilate to release heat, and sweat glands produce sweat that cools the body through evaporation. When the body is cold, blood vessels constrict to conserve heat. Melanoma can disrupt this process in several ways. Tumor invasion can damage dermal blood vessels, making it harder for the skin to dilate and release heat. If sweat glands are destroyed, the body loses some of its ability to cool itself through evaporation. Inflammation around the tumor can also alter blood flow. And after surgical removal of melanoma, the loss of glands or vascular structures can further reduce thermoregulation efficiency. To conclude this presentation, it's important to recognize how the normal structure and function of the skin play a vital role in protecting the body and maintaining homeostasis. Melanoma begins in the melanocytes of the stratum basale, and in many cases, like the one in our scenario, long-term ultraviolet exposure is the primary cause. As melanoma progresses, it doesn't just affect the epidermis. It can invade the dermis, disrupt accessory structures, and even extend into the hypodermis. These changes compromise essential functions such as thermoregulation, immune defense, and tissue integrity. This case highlights why early detection is so critical. When melanoma is identified before it penetrates deeper layers, the chances of successful treatment are significantly higher. Understanding both the anatomy of the skin and the pathology of melanoma helps us better appreciate the importance of prevention, screening, and timely medical intervention.